Hi, my name is Róisín McManus. I'm a research group leader at the DZNE here in Bonn. I'm interested in microglia and the role that they have in the development of neurodegenerative diseases, including Alzheimer's disease and dementia. I'm first author on the paper entitled NLRP3-mediated glutaminolysis controls microglial phagocytosis to promote Alzheimer's disease progression that was just published in Immunity. And in the next 2 to 3 minutes, I'm going to tell you all about our findings. In our paper, we found a new role for NLRP3 outside of its inflammasome complex, where it can regulate microglial metabolism and the metabolites being generated, which had a big influence on the genes being transcribed, thus reprogramming the cells to make them very efficient at phagocytosis. Previously, in microglial cells, it was thought that NLRP3 can only function inside of its inflammasome complex, and we discovered something very different. Under resting conditions, NLRP3 was actually associating with the mitochondria and microglia. When we used pharmacological inhibition to block this, we found that the microglia were increasing their use of the nutrient glutamine and producing more of the metabolite alpha-ketoglutarate. This is a very interesting metabolite because it has another function in cells where it can affect the epigenetic landscape, which means how open or closed the DNA region is around certain genes. In our experiments, when we block NLRP3 and increase alpha-ketoglutarate, the region around certain phagocytosis genes was more open and active, allowing these genes to be transcribed. This reprogrammed the microglial cells, where they became very good and efficient at phagocytosis. In Alzheimer's disease, we know that microglial phagocytosis can become impaired, and so we asked whether our mechanism would be of relevance. We found that when we inhibit NLRP3, boost alpha-ketoglutarate and turn on these phagocytosis genes, microglia are very efficient at phagocytosing amyloid beta, and this was protective in our models of Alzheimer's disease. Our findings show that there are two roles that NLRP3 has in microglial cells. One is the inflammasome complex, which has been very well characterized. And our new finding shows that NLRP3 can also control microglial metabolism and how it's using nutrients like glutamine. This is really important because it highlights NLRP3 as a really important therapeutic target in dementia. It also shows that microglial metabolism and the specific metabolites generated have a really critical role on cell function. And this could lead to future therapies moving forward targeting the immunometabolic pathways. My favorite figure is figure 6, because during the process of uncovering this pathway, we had initially used microglia deficient for NLRP3, which showed a lot of these metabolic phagocytic differences. But using the NLRP3 inhibitors, I was able to eventually work out the mechanism behind this pathway. It actually took a very long time to establish the time points, but I was ultimately able to uncover the sequence of events, and a lot of that data is in figure 6. Following on from our discovery that NLRP3 can control microglial metabolism and specific metabolites, in my lab we're now working to better understand the role that different metabolites have on microglial function in health and disease. We're also trying to translate our findings and see if they're relevant for other neurodegenerative diseases. And moving forward, we think that targeting immunometabolism beyond NLRP3 could be a really promising therapeutic treatment for those with Alzheimer's disease or other dementias.